Hello friends, today we are going to discuss the membrane proteins. The plasma membrane consists of phospholipid bilayer with associated proteins. The proteins are the major constituents of plasma membrane, making between 25 to 75% of the mass of the different membranes. The membrane proteins are amphipathic molecules in which hydrophilic portion interacts with the aqueous environment on either side of the plasma membrane, and the hydrophobic portion interacts with the phospholipid bilayer. The proteins can be anchored to membrane by lipids covalently linked with the polypeptide chains. The cytosolic proteins are attached to membrane by 14-carbon myristic acid attached to their N terminus. Some may use 16-carbon palmitic acid or 15 to 16-carbon prenyl groups attached to cysteine side chain for anchoring to the plasma membrane. The phospholipids attached to C-terminus of proteins anchors them to the extracellular leaflet of the plasma membrane. According to the Singer and Nicholson's fluid mosaic model, proteins float in the sea of phospholipids like mosaics. The membrane proteins perform specific functions like anchoring, reception, transportation, etc. The membrane proteins are divided into two basic types, that is the integral membrane proteins and the peripheral membrane proteins. The integral membrane proteins are firmly associated with the plasma membrane. They can be separated with agents like detergents such as SDS or Triton X100, organic solvents or denaturants that interfere with hydrophobic interactions of the membrane lipids. The integral membrane proteins in the form of transmembrane proteins serve as channels or carriers or the pumps during transmembrane movements. Most integral membrane proteins called transmembrane proteins have a stretch of about 20 to 25 non-polar or hydrophobic amino acids that span the plasma membrane. Porins present in the outer membrane of some bacteria, chloroplast, and mitochondria contains beta barrels that are the barrel-like structures formed as a result of folding of beta sheets upon itself. The integral membrane proteins may have single or multiple membrane spanning domains. The integral membrane proteins exhibit asymmetry orientation that enables functional asymmetry. The transmembrane proteins have tyrosine and tryptophan residues at their lipid-water interface. The alpha helix and beta sheets present in the transmembrane domain of the integral membrane proteins are stabilized by the intra-chain hydrogen bonding and the hydrophobic interactions. Second type of membrane proteins are the peripheral membrane proteins. The peripheral membrane proteins are weakly associated with plasma membrane through electrostatic interactions or hydrogen bonding with hydrophilic domains of either integral membrane proteins or the polar heads of the membrane lipids. These can be dissociated from plasma membrane by treatment with polar agents like carbonate at high pH or high salt concentrations that interfere with electrostatic interaction or hydrogen bonding. Peripheral membrane proteins serve as anchors for membrane-bound enzymes and limit mobility of integral membrane proteins. The peripheral membrane proteins, once dissociated from plasma membrane, are soluble in aqueous environment. Thank you.